Welcome to the deep dive. Today, we're getting into something pretty fundamental, really a paradox of life. How cells are programmed to die. We're talking about apoptosis.
All right, programmed cell death.
Exactly. Now, when you think cell death, maybe you picture, I don't know, a car crash, something messy. That's necrosis.
Yeah, necrosis is messy. The cell membrane breaks, everything spills out, causes inflammation. It's an uncontrolled demolition.
But apoptosis, that's different.
Totally different. It's, well, elegant almost.
Yeah.
The cell shrinks, the insides condense, and crucially, no inflammation. It's clean. Vital for health, actually.
Essential for everything, from how we develop as embryos to getting rid of cells that might turn cancerous. It's like the body's quality control.
Precisely.
So our mission today, unpack the machinery, the molecular nuts and bolts that decide if a cell lives or dies. We need to map out the two main ways this happens.
Yeah, there are two major routes, and understanding them is key for understanding diseases like cancer or autoimmunity. You've got the intrinsic pathway that's triggered from inside the cell. Think internal damage alarms.
Right.
And then there's the extrinsic pathway, which gets its signal from the outside, like an order coming in, often from the immune system.
Okay, let's start with the inside job then, the intrinsic pathway. What kind of internal alarms set this off?
We're talking serious cellular stress, things the cell just can't fix.
Like what specifically?
Well, major DNA damage is a big one, say from too much radiation or certain toxins, or maybe hypoxia, not enough oxygen. The cell basically senses, okay, I'm too damaged, I could become dangerous.
So it decides to take one for the team.
Kind of, yeah. And that decision point, the real battleground, is the mitochondrial outer membrane.
Ah, the powerhouse of the cell.
Right, but here it's acting as a gatekeeper. And there's this constant push and pull involving the BCL2 protein family.
Okay, tell me about them.
So you've got the pro-survival guys, BCL2 itself and BCL2-XL. They're the stabilizers. Their job is to keep that mitochondrial membrane intact, specifically by stopping pores from forming.
They're like the security guards on the membrane.
Exactly. And then you have the ones trying to breach the walls, the pro-apoptotic proteins, BX and BA.
The poor formers.
Yep. When that stress signal hits, like the DNA damage we mentioned, BX and beta get activated, they actually move to the mitochondria and start clumping together.
To do what? Punch holes.
Essentially, yes. They form these channels, or pores, in the outer membrane. This whole process is called MOMP.
Mitochondrial outer membrane permeabilization. Catchy.
Ah, yeah. It means the gates are breached.
And you mentioned DNA damage. Isn't there a famous protein involved there?
Ah, yes. We have to talk about P53, often called the guardian of the genome.
Right.
So P53 is constantly checking the DNA. If it finds severe damage that can't be repaired, it acts as a potent tumor suppressor.
Oh, by fixing it.
Sometimes. But if it's too bad, P53's job is actually to trigger apoptosis. And it does this crucially by upregulating BX.
So P53 tells BAX, go make some holes in the mitochondria.
That's basically it. It activates the demolition crew.
Which explains why defects in P53 are so linked to cancer.
Absolutely. Think about Li-Fraumeni syndrome. People with this have a faulty TP53 gene from birth. Their cells can't properly activate BX when damaged. So damaged cells survive, replicate, and their cancer risk skyrockets. The intrinsic pathway's emergency brake is faulty.
Okay, so BAX and BAK make the pores.
Yep.
MMP happens.
Yep.
What comes out?
The key molecule released is cytochrome C.
Wait, cytochrome C, isn't that involved in making energy cellular respiration?
Normally, yes. It's essential for energy production inside the mitochondria, but when it leaks out into the cytosol, the main cell fluid, it becomes a death signal.
Wow, talk about a career change. From energy production to execution trigger.
It's pretty dramatic, yeah.
So cytochrome C is loose in the cytosol. The alarm's gone off. What happens next? How does this one molecule cause total breakdown?
Well, cytochrome C doesn't act alone. It needs a platform, an assembly point.
Okay.
It finds a protein called a PAF1 that stands for apoptotic protease activating factor 1. Think of it as scaffolding. Cytochrome C, a PAF1, and some ATP cellular energy link up. And they form this large, rather intricate structure. It looks like a 7-spoke wheel. It's called the apoptosome.
The apoptosome, a death machine, basically.
Pretty much. Its sole purpose is to kickstart the real demolition crew. The caspases. This starts the caspase cascade.
Caspases, those are enzymes, right?
Proteases.
Exactly.
Protein-cutting enzymes. The apoptosome specifically activates the first one in this intrinsic pathway chain. caspase-9.
Initiator caspase.
Right. Caspase-9 is like the general receiving the orders from the apoptosome. It doesn't do the bulk of the destruction itself.
What does it do then?
Its job is to activate the troops,
Yeah.
the executioner caspases. It activates them in huge numbers.
And who are they?
The main ones are caspase-3, caspase-6, and caspase-7. But caspase-3, that's the real workhorse here.
The molecular demolition man, someone called it.
Huh. Yeah, that fits. Once caspase-3 is active, it's relentless. It starts chopping up crucial cellular components, like things like the cytoskeleton proteins. That's why the cell shrinks and loses its shape. It targets the proteins holding the nucleus together, the nuclear lamina, so the nucleus fragments. It also cuts the proteins that help cells stick together, so the dying cell detaches cleanly.
Wow. It's incredibly systematic. A controlled implosion.
Exactly. Once caspase-9 fires up, that cell is committed. There's no turning back.
Okay, that intrinsic pathway makes sense, the cell policing itself. But what about the extrinsic pathway, when the kill order comes from outside?
Right, so this is often initiated by the immune system. Say, a cytotoxic T cell finds a virally infected cell or maybe a precancerous one.
And it needs to tell that cell time to go.
Precisely. It does this using specific signals called death ligands.
Ligands, like keys fitting into locks on the cell surface.
Exactly. These ligands bind to specific death receptors on the target cell's plasma membrane. Classic examples are Fas ligand, or FASL, binding to its receptor, Fas also called CD95, or another one is TNF-alpha binding to its receptor, TNFR.
The killer handshake.
That's a good way to put it. Highly specific.
So the handshake happens on the outside. What does that trigger inside the cell?
That binding event causes the receptors on the inside part of the membrane to cluster together. This recruits an adapter protein called FADD.
FADD, fast-associated death domain.
Right, FADD is the bridge. It then recruits multiple copies of an inactive caspase procaspase 8.
Okay.
This whole cluster, the receptor FADD and procaspase 8, forms another complex. This one's called the DISC.
DISC, death-inducing signaling complex. Sounds ominous.
It is. The DISC functions similarly to the apoptosome in the intrinsic pathway. It's the activation platform.
And it activates.
It activates caspase 8, the initiator caspase for the extrinsic pathway.
Ah, so the intrinsic pathway uses caspase-9 as the initiator, and the extrinsic uses caspase-8.
Correct. But here's the interesting part. Both caspase-9 and caspase-8, once activated, ultimately lead to the activation of the same executioner caspases.
Like caspase-3 again.
Exactly. They converge on the same final execution machinery.
So why have two separate pathways, caspase-8 and caspase-9, if they end up doing the same thing, activating caspase-3? What's the point?
That's a great question. It boils down to redundancy and specialization.
Yeah.
Having two routes makes the system more robust. You can't easily escape death just by blocking one pathway.
Makes sense.
And they handle different situations. The intrinsic pathway is for internal problems, damage, stress. The extrinsic pathway is more for, well, external threats or commands, often from the immune system clearing out infected or unwanted cells quickly. It allows for targeted removal without waiting for the cell to damage itself first.
Two different triggers converging on the same demolition crew. Okay, so caspase-3 gets activated, starts chewing things up. You mentioned the nucleus fragments.
Mhm.
What about the DNA itself? Does it just get shredded randomly?
No, not at all. This is another hallmark of apoptosis, incredibly precise. Caspase-3 activates specific DNA-cutting enzymes called endonucleases. And these enzymes don't just chop randomly. They cut the DNA very specifically in the linker regions between the nucleosomes, the protein spools the DNA is wrapped around.
Like cutting a string of beads exactly between the beads.
Perfect analogy. Because the DNA between the nucleosomes is a somewhat consistent length, you get DNA fragments of specific sizes.
And how do we see that?
If you take the DNA from these dying cells and run it on a gel electrophoresis, you don't get a random smear like you would in necrosis. Instead, you see a distinct pattern.
A ladder.
Exactly. A DNA laddering pattern with fragments typically around 180 base pairs apart and multiples of that. It's like a molecular fingerprint for apoptosis. Very clean, very specific.
That's amazing. Such control right down to the DNA level, unlike the messy smear of necrosis.
It highlights the program nature of it.
Yeah.
But when this program goes wrong, well, that's where you see significant disease.
Right. Let's talk clinical consequences. What happens if, say, those anti-apoptotic proteins, BCL2 or BCL-DAXL, are too active?
If they're overexpressed, they effectively jam the mitochondrial gates shut. BAX and BA can't form pores. Cytochrome C doesn't get released.
So the intrinsic pathway is blocked. The cell becomes resistant to dying.
Precisely. And that's a major mechanism for cancer cells to survive and resist chemotherapy. The chemo might cause DNA damage, which should trigger P53 and BAX.
But if BCL2 is holding the fort, the signal gets ignored.
Exactly. A classic example, really textbook, is follicular lymphoma.
Okay, what happens there?
In many cases, there's a specific genetic mistake, a translocation, written as T1418. This swaps pieces of chromosome 14 and 18.
And the result?
The result is that the BCL2 gene, which makes that anti-apoptotic BCL2 protein, gets moved next to a really strong, constantly active promoter sequence, usually used for making antibody genes.
So instead of normal levels of BCL2, the cell is just churning it out constantly at high levels.
Massively overexpressing it. So these lymphoma cells are incredibly resistant to apoptosis. They just refuse to die, even when they should. That's central to the disease.
Wow, a single gene placement causing so much trouble. What about problems with the extrinsic pathway?
Defects there can cause significant issues too, particularly with the immune system. There's a condition called ALPS.
ALPS, autoimmune lymphoproliferative syndrome.
Right. This often results from mutations in the gene for either the FAS receptor or its ligand, FASL.
The killer handshake components.
Exactly. Normally, the FAS system is crucial for getting rid of immune cells, particularly T cells, after an infection is cleared, or for eliminating T cells that might attack the body's own tissues, autoreactive cells.
So it's a cleanup mechanism for the immune system.
It is. But if FAS signaling is broken because the receptor or ligand is faulty, those autoreactive T cells don't get the death signal. They don't undergo apoptosis.
So they just hang around.
They hang around, they accumulate. This leads to the lymphoproliferative part, swollen lymph nodes, and large spleen. And because they're often autoreactive, they can start attacking healthy cells, causing autoimmune problems like low blood cell counts called cytopenias, all because the extrinsic off switch for those immune cells is broken.
So failures in either pathway have really serious consequences. Cancer on one side, autoimmunity on the other.
It really underscores how critical this balance is. Apoptosis is this fundamental quality control. You need that constant tension between the pro-death signals like VX and BAK and the pro-survival ones like BCL2 and BCLSL, ready to tip the balance when a cell needs to be removed.
It completely changes how you think about health and disease at the cellular level. Okay, so we know things like, say, radiation causes DNA damage, triggering P53, then BAX leading to apoptosis. That's the plan.
Right, the programmed response.
But thinking about something like that follicular lymphoma example where BCL2 is way too high, blocking that intrinsic path. Here's a final thought for you listening. If a cell has essentially disabled its primary self-destruct sequence, its programmed apoptotic pathway, how else might it die? Could the body or the environment force its hand? Maybe push it towards a different, less controlled death? Perhaps overwhelming it with something like free radical damage, the kind you see in ischemia reperfusion injury. Could that bypass the blocked apoptosis and essentially force the cell into that messy necrosis we started with?
That's a fascinating question. Exploring those backup mechanisms, or maybe the lack thereof, it really shows the complexity and the potential vulnerabilities in cell fate decisions.
Definitely something to chew on. Thank you for joining us for this deep dive into the intricate world of apoptosis. We hope you feel up to speed. Catch you next time.